is at the end of an era for strong government support of biomedical research. It's Eric Green next on the show. Welcome to Mendelspot, everyone. I'm Theral Timpson. Eric Green served as director of the National Human Genome Research Institute for more than 15 years. Before that, he spent nearly 2 decades in leadership roles inside NHGRI as scientific director, head of its genome technology branch, and founding director of the NIH Sequencing Center after helping to drive the original Human Genome Project. Out of 31 years at NIH, Eric has just told me he was in leadership position for 29 of them. Under his leadership, NHGRI launched critical initiatives, sprawling strategic plans that shaped the course of genomic medicine, major efforts to decode genome function, to enable genomic data science, and to apply genomics to diagnostics and rare disease and precision medicine. He's a big champion of precision medicine. In recognition of that work, Green has received numerous honors, including election to the National Academy of Medicine in 2023. And just in October of this year, ASHG honored him with the American Society of Human Genetics Leadership Award. And Eric has authored nearly 400 scientific papers that have helped define our field. But now, after a sudden and unexplained non-renewal of his appointment earlier this year, Eric finds himself in a new chapter outside the corridors of power, yet still deeply connected to the future of genomics. Eric Green joins us now. Welcome back to the show.
Well, thanks. Thanks for having me. It's always great speaking with you.
Congratulations on this ASHG award.
Oh, thank you. It was very flattering, very humbling. It really meant a lot to me. And the award was given to me, but it was as much a reflection of the contributions of the National Human Genome Research Institute to the field of genomics as anything else. And, I was there for 31 years professionally and came in as a junior investigator and left as the director, and not by my choice, but being honored in that way really is really so remarkable accolades that go to the Institute more broadly.
So before we talk science and policy, I want to keep it personal. And, obviously that chapter suddenly ended earlier this year for you. I want to ask, how are you? What has this time been like for you personally?
It was unexpected. To be honest with you, I only was given about 2 or 3 days notice that I was going to have to retire from federal service. I was fully retirement eligible. I had a reappointment date. I had been reviewed, which is the step before you get reappointed, and was reviewed very positively. All the paperwork was done. It was just literally a switch in a computer. I had to go in and just say, reappoint him. And nobody told me that wasn't going to happen until 3 days before, where they say it might not happen. And by the way, when it did happen, I was forced to retire, and I was retirement eligible. And I sometimes tell people, when you're in government service for a long time, one of the things you do get is a pretty good retirement package, but it's complicated. And in fact, what most people do when they're getting near the end of their government career is they go take a class about how to retire from federal service so that you maximize. You got a lot of decisions to make.
And you hadn't taken the class.
Oh my God. But by the way, not only do you get to take the class, the government, which sometimes isn't particularly generous, will pay for your partner to take the class with you so that as a couple, you can make all the decisions. And so my wife and I never took that class. We had talked about it and said, well, maybe in the next 3-4 years, we should take this class because at some point I'm going to retire. But, I did not even spell retirement. But all of a sudden, on 3 days' notice, I was forced to retire from federal service. We got the class. They gave us an HR person by Zoom for about an hour and a half on a Sunday afternoon. But like a 1-week class became condensed into an hour and a half. And I retired from federal service, but as I've said repeatedly, and to answer your question, how I'm doing, I don't consider myself retired. I'm very busy. I'm doing lots of things. I haven't latched on to a permanent position, and may or may not do that. We'll see. But
you don't know. Okay. All right.
I don't know exactly at this moment, but I am exploring multiple options, and there will be, I don't know, version 1.0 was Eric as an academician, and version 2.0 was Eric in the government. There will be a version 3.0.
That's very resilient. of you. And, I think scientists across the country are feeling somewhat similar to what you've just lived through, a sense that the ground beneath American science has cracked.
Wow.
How
It's
do you
really gotten
feel
very
about
squishy. Yeah, it's really bad.
it's? Yeah, I mean, talk about that. And scientists, what was the mood like at NHGRI?
Oh, well, I mean, there's so many comments I can make. First of all, NHGRI specifically, I mean, they were orphaned. I mean, they did like me, I think most of them, and they got orphaned. And then a week later, the deputy director, who was the logical person to be the acting director, a wonderful, talented scientist named Vince Bonham, he was terminated. And not because he wasn't reappointed, he was terminated a week later. So they lost their director, they lost their deputy director. And meanwhile, this was in March. By April 1st, which was the horrible day at NIH, was when the reduction in force happened. That's the RIF, but they sometimes call it being rift. And that's
Yes.
when major groups of people got terminated, or they got put on administrative leave. They eventually got terminated about 6 or 7 months later. But our entire communications group, our entire education group, our entire policy group, our entire workforce development group, that was the saddest day I've ever been at NIH. I actually wasn't working there anymore, but I was still going to work just to sort of provide emotional support and to advise people. And all of a sudden, surrounding me, 35, 40 people who were the closest people I were working with, all the people with offices immediately around me, they all got notices that they were being terminated. So trauma is the word I would use
Trauma.
for the
Yeah.
institute. And
And
on
what was
top of it, of course, there was also incentivized retirements and people that were probationary employees that got terminated earlier. So this was this cascade of things that went on throughout March, even starting in February, of losing staff. I mean, the institutes, on inauguration day, I was overseeing the institute of 650 people, and well over 100 people are now gone. And you're seeing great colleagues that are just separated from you.
I've seen pictures on like PBS NewsHour of labs inside the NIH that are sort of just decimated. Things in the freezer going bad. So what kind of an interruption has this been for science?
I don't think we completely know the answer. It's been very disruptive. So first of all, yes, it's been incredibly disruptive, and there's studies that are out there of how many clinical trials have been canceled, delayed, how many patients have been affected. So there's a lot, but I don't think we know the full depth of the damage, in part because the damage is still happening. We're not done. There's still news every week or two, unfortunately, of more bad things happening at NIH or at CDC or at FDA. And so we're not at the end yet. You gave the example maybe of seeing something on PBS and within our on-campus, or we call our intramural program. And yes, that has been problematic because people have lost staff. You can't replace staff. There's lots of insecurity about many things. But in many ways, the intramural program was spared more because they really didn't fire that many active intramural scientists or staff, except for the ones that were probationary employees who took early retirement or things like that. But it was major groups like communications groups and education groups, policy groups, so forth. They're just gone. I mean, these institutes just don't have them anymore. I can tell you that our website, genome.gov, which is one of the most heavily used websites that inform people about genomics and genomic medicine and so forth, it hasn't been updated since April 1st because there's no staff to update it. And there's all sorts of incredible good outreach, education, communication stuff on genomics to help people in genomic literacy that's just sitting there. And, it's not being updated. It's not being, if links get broken, they can't get fixed right away. I mean, it's a problem. It's going to decay over time. And the consequences of that are going to be hard to measure.
I've heard that most of the funds are now going out.
Yes.
I saw, I think it was New York Times showed that they were like paying full funds off just to spend money, like 4-year funds rather than a year at a time. So some are being shorted.
It's a concept called multi-year funding. The way most NIH grants go out is they go out where, let's say they got a 5-year grant, you get 1/5 the first year, a second fifth the second year, and so forth, and you parcel it out 1 year at a time. And there is a thing you can do that was done very selectively called multi-year funding, where if you happen to have all the money for all 5 years, you give the university all the money at once. And that's a very useful tool. I used it occasionally when you got to the end of the fiscal year. You didn't really have very many grants you wanted to fund, maybe one more. You had all the money for it. You gave them all the money because then you didn't have to think about them for 4 years. They were set. You didn't have to budget for them for the next 4 years. But what happened last year was that a good tool of multi-year funding, which I think is a good tool, needed to be used in a bad situation. And that caused some really problematic situations. Because what happened was that there weren't enough staff to get the grants out the door because of all the terminations and all the probationary firings and all of the early retirements and all that stuff. So at the end of the year, and plus there were all the delays caused by all the things that happened early in the year. So, but when September 30th was arriving, which is when every dollar has to be spent, there were not enough people to get enough grants out the door. So rather than giving 5 grants, which was a lot of work, they would give 1 grant, but they would fund it multi-year.
And fully funded.
Now, that was great for that one investigator who was then funded for 5 years. But the other 4 investigators who didn't get their funding at all, they were completely out of luck. And if they needed that for a technician or somebody in their lab, they probably had to terminate that person because that money wasn't going to come in. So again, as a result, multi-year funding got a bad name and was sort of said, oh, this is a horrible thing. It shouldn't go on. The only reason it was a horrible thing was because we were in a horrible situation and it had this bad consequence. Used in a good situation, it's a wonderful tool.
Now, Trump suggested a 40% cut to the budget. The House and Senate have not gone along with that so far. What do you think?
If there's a silver lining of 2025,
yeah,
you gotta, there is silver linings in 2025. They're just few and far between, and you gotta get it away from all the horrible things. Bipartisan support once again for NIH, and it's so rewarding to see that. Unanimous Republican support out of both appropriations committees for NIH. They would have no part of a 40% reduction. In fact, they basically came in at flat. And so while there's still lots of problems that NIH is facing, it doesn't seem that budget is one of them. At least the overall aggregate budget is going to be the same. I mean, it'd be better if it was up, but under the circumstances, I think just having it be flat is far better than a 40% reduction, which would have decimated the biomedical ecosystem in the United States, to be honest with you.
Yeah, set us way back behind other countries.
Even a 10% cut would be horribly detrimental. 20%, just keep going. I mean, 40%, it would have been a total calamity.
The irony here is the last time you were on, we titled it Eric Green on the Future of the NHGRI.
There still would be a future of the NHGRI.
Okay, so let's talk about that. Because one of the big problems for the Trump administration has been diversity initiatives. And it's one of the things that was in your strategic planning that we discussed last time. And we're talking diversity of genomic databases, which is, there's science in that. Also diversification of the workforce, which there's science in that too. Talk about your sense on those goals and where we're at.
So I think it's a gradient. I mean, first of all, the battles are still going to happen. I mean, they're happening now. They started before I was terminated, and they're going to continue. And I really like the way you framed it, because when when you talk about diversity, you talk about DEI, whatever phraseology you want to use, context is everything, especially in the genomics context of diversity. And so one kind of diversity is about the data. And to me, this is a really important distinction. And I think the administration is going to be okay on this. Any of our programs related to diverse genomic data have not been threatened. And the reason, just so your listeners fully appreciate it, the reason we need to collect a lot of data, of your genomic data, mostly genome sequence data, from people from all over the world, from all different ancestries. Yes, they are, you could use the diversity word, but the most important thing is they just incredibly heterogeneous data from people from all different histories of their ancestry. The reason for that is that data is used to interpret a genome sequence of a patient in order to help establish what the genomic differences are, the spelling differences in that patient's genome. And you must have properly matched control data from somebody of similar ancestry in order to make sure what you get out at the other end is accurate. Otherwise, you make mistakes. So in other words, if you're analyzing a patient of one ancestry and your comparator data is of another ancestry, there will be errors, there will be omissions, and studies have shown this.
Let's take an example, because some populations are more prone, say, to heart disease. Can you give us an example of this where that population may not have been studied? For in the beginning, it was mostly white Northern European in the databases.
Well, actually, you're raising a whole other issue. Is the other reason why you want to have genomics being studied in all different ancestries is for people with specific diseases so that you can get to the spelling differences. But the point is, what I was talking about isn't even genomic data from individuals with a given disease. It's just baseline genomic data of somebody from Thailand or something like that.
Yeah.
And that person, when you have a patient who is Thai ancestry, you don't want to compare their genome sequence to a genome sequence like mine, because we're way too different. We're actually mostly the same, but there are enough differences that to make it accurate, you really want to have the comparator be somebody from the similar ancestry. And by the way, the reason this is so medically important in the United States is think about the US, this great melting pot with so many people of mixed ancestries. If we don't have heterogeneous genomic data, notice I use the word heterogeneous rather than diverse, but heterogeneous genomic
All right.
data.
Maybe I should start doing that on the program.
Yeah, well, it just keeps me out of trouble. By having that, we will be able to take individuals, anybody in the US, and we will be able to, in the long run, be able to more accurately analyze their genomes, because we'll have available the comparators that include the people, who are of similar ancestry as them.
Okay, so but your sense is the administration understands that on this point. Right. Yeah, so that they can block the border and stop immigration and stop diversity that way. But to do it with science is just to stop doing basic science.
Well, but remember, genomics is finding its way into the clinic. This isn't just for good science. This is going to be for good medicine. Yeah. When people are going to be getting genomic testing done as part of medical care, if we don't have good comparators, we will make medical mistakes. And there's literature describing this, where people are missing things because they ended up not analyzing the patient's genomic data relative to the right comparator, the comparative data. So I think we're okay there, but we need to constantly tell people, this is not about a social reason. This is not about, this is for medical purposes. We need to have it available. The second thing you mentioned was workforce. And this gets a little more subtle. And I think we're going to have to really battle this, because the situation is that genomics carries with it a little bit of it carries with it real history of eugenics. And eugenics disproportionately marginalize some individuals from certain ancestral groups compared to others. Oh, and by the way, certain individual groups are more distrustful of medicine in general. So anytime you bring them something new, they're a little distrustful to want to participate in biomedical research. So I would contend that the combination of those things, that as we implement genomics into clinical care and we approach more and more patients of, we would like your permission to sequence your genome to be able to most effectively treat you medically, will you give your consent to allow that to be done? Or we would like you to participate in this research study. Would you consent to have that done? I would contend that especially with certain communities, such as communities of color, where there have been missteps in biomedical research, and thinking a little bit about eugenics in general, that we have an extra burden on our back in order to get trust and engagement with all communities to participate in genomics for research and also genomics for clinical care. And as a result, I would contend that if all the people working in genomics look like me, we may not be trusted by some communities. We need members of those communities to be the genomicists, to help be the research coordinators and the nurses and the doctors and the pharmacists who are going to go in there for both research and for clinical care and say, hey, look, you're more likely to trust me because I come from a similar group as you. And we believe, and I think science has shown this, that that is more helpful than just having a non-diverse workforce, which generally would end up being a lot of people that look like me and you. And so
Yeah.
when you start developing programs that are going to enrich for individuals from underrepresented groups in genomics, there's some concern around that in the current administration. And so that we have some programs that we launched that are getting some limitations around it or being modified. So I think that's a work in progress.
Argument well made there. I thought you put that super well.
By the way,
I remember
the third category, which I think is very much under threat, is one of the things that happened that I'm very proud of. And I was very proud of it throughout the time I was the director, were programs we had also for building capacity in genomics outside the United States. And the exemplar project for that was a project called Human Heredity and Health in Africa, or H3 Africa. This was a program that we did jointly between the NIH and the Wellcome Trust in the UK to basically build capacity for genetics and genomics research on the African continent to try to help Africa catch up to the genomic revolution. And it was a very successful program. And there's a lot of continuation of some aspects of things we started. A program like that would almost be impossible to start now. And there are other things we were working on in the previous administration of potentially doing something similar in other parts of the world. And all of those ideas are pretty much have to be put on ice right now. I should be put in cold ice. I've got it for. I apologize for that. It needs to be put in cold storage for now, because those ideas will just not get root in the current environment that we're in. But nonetheless, I would just say I think it's very important that we take a world view to thinking about how to build genomics around the world, as the U.S. has always been so effective at improving education and capabilities and technologies to help everybody around the world.
Well, a few years ago, we were talking about pandemic preparedness and new initiatives, right, to get better ahead of that the next time that happens. So yeah,
well,
I hear
I'm not
you.
the expert on pandemic preparedness, but I know enough to say that we're falling behind again.
Is there anything in the new way of thinking, the new administration's arguments, that has changed your mind or made you think double about something?
Well, what it has done is it has forced me. I mean, just thinking about the conversation we were just having, is I would just tend to, because I'm sort of have this altruistic, metamorphosis in genomics that we share everything. We try to get everybody involved. We try to be inclusive. I mean, to me, that's just part of my heart and soul. It's probably, buried in my genome deeply. But what I've had to do is to think about where they're coming from and then try to parse out these ideas of these different elements of diversity to figure out what will they agree with me on, what were the things we're going to have to argue about, and what are the things that are just off the table. So I guess
so it's kind of all bundled together in a sense. But to tease that,
bundle it all together because it was all bundled under altruism, and altruism isn't just part of what we can be thinking about now. So you got to be a little more nuanced and really think about how you separate some of these things and asking whys around each of them.
I think that's some wisdom for researchers listening to the show. Just be aware of how you frame things.
But some of this is silly because some of the researchers have found themselves having to, get away from what they call, the bad words and the naughty words. If you have a yeast geneticist who would, the poor yeast geneticist, there's many stories like this, where in their grant, they talked about, I'm going to use a diverse set of yeast strains for this nerdy experiment I'm going to do. And their grant got canceled. Their grant got canceled.
That sounds like an AI program.
Yeah. Then they got reinstated and they said, oh no, but they only look for diversity. So now everybody says, I'm going to use, a large assortment of different yeast strains. And so you avoid the D word. But that's just silly, right? That shows it's just silly.
We'll look back on that and say, wow, did we live through that? Let's pull the camera back a little and just talk about the field of genomics today. And maybe a little bit more the scientific direction. It feels so rich. We're sequencing for $100. We're getting human genomes for rare disease in under 24 hours. Roche has this new sequencer out. The technology is still evolving. And now it's become more fragmented with like multi-omics, single cell, spatial, Long reads. We did a program on this a couple weeks ago.
Yeah,
Long reads are making it into the clinic.
yeah.
And then AI, has come along and changed. So where do you think we
You've
actually?
obviously heard one of my recent talks. You're hitting all the highlights.
Oh, no, I missed it. I missed it. Where do you think we actually stand as a field right now? I know that's a big question, but
Yeah.
how, looking back over 15 years, where did we make real progress, and where could have we done better, or what is some of the noise?
Hearing you rattle off those highlights, it really amplifies this idea. What a whiplash existence we have right now. I mean, talk about the best of times, the worst of times. I mean, what's happening, with all this political stuff and NIH and, me personally, but lots of others in the field, we're dealing with just some of this is like the worst year in the world. It just sucks. I mean, there's no way to say it. And yet, if you just ignore that for a minute, or you go to the American Society of Human Genetics meeting, or you go to a precision health meeting I went to, or, I was just at the Mexican, Society of Human Genetics Congress. What an incredible opportunity and current accomplishments happening in genomics truly is mind-boggling. And one of the things I give a lot of talks. I mean, and even now, even no longer at NHGRI, I'm constantly giving talks, constantly preparing talks. And sometimes I give a talk where, like, I'm going back, like I just gave a lecture in a class every year, and I give the same lecture, except I update it every year because it's all about the latest advances in genomic medicine. I can't believe how many new slides I had to put in the version I just gave yesterday compared to a year ago. In fact, I made a whole new slide about all these incredible therapies. And by the way, I have no expertise in genomic therapies. NHGRI has done very little in the therapeutic space. Most of the other institutes have done that. But I mean, there's all these genome editing, there's gene therapy, there's sickle cell, there's small molecules for cystic fibrosis. There's cell therapies. It is just incredible. We are truly now bringing the diagnostics of genomics into the therapeutics, and it's just mind-boggling. But your list of highlights are everything from the most basic, still developing new technologies, the new Roche machine, the new other technologies, long-read sequencing. Once upon a time was going to be for specialists. It's going to find its way into clinical care. AI intersecting with all the complexities of interpreting genome sequence in terms of function, but also understanding genomic variation. The list just goes on and on. I would say one of the big things I've added this year to my talks that I've gotten very excited about, perhaps anticipating a question you may ask me at an end of year time, what have I got? I've really gotten excited about this idea of newborn sequencing, genome sequencing of all healthy newborns. Previously,
okay, great, yeah. That's very interesting because we had Catherine.
We are in a phase change going on now, and all you got to do is follow the news. I've always thought that there will be some day where most newborns will get their genome sequenced. I'm now changing this in part because I'm following the news. What's in the news? Well, more and more and more countries are doing major studies where they're piloting the routine genome sequencing of healthy newborns and integrating that into their medical record and figuring out how it is better than the screening that's done for a few dozen genetic disorders, which most healthy newborns get in over 100 countries in the United States. And that includes, even in the United States, 2 weeks ago, NIH just releasing a new program or launching a new program called Beacon. But there's other US programs. But the thing that really caught my eye was earlier this summer, the UK, which I think is leading the world in genomic medicine implementation, largely in part because they have a single payer system, so it's
exactly
easier to do the studies.
And that's been the case for a while.
And they're doing a massive program in newborn sequencing tens of thousands of healthy newborns and integrating into their health system, et cetera, et cetera. Well, they came out earlier this summer and just declared within 10 years, they believe that every baby will get their genome sequenced at birth. So they sort of put a marker down. 10 years is not that long of time to prepare for something that seismic. And, I see what's happening here in the US. Examples, you got to follow the news and genomics, which people like me do. But, Florida just passed a new state law. I mean, Florida is not the most progressive state in the union. And
no, but it's truly some good news this year.
Genetic Sunshine Act. in Florida, where they're starting to do much more exploring. Well, they're first of all doing a sequencing of acutely ill newborns, but they're exploring much more aggressively the idea of healthy newborns. This is catching on. More and more states. This new Beacons program out of NIH. The other studies that have been going on there's a lot of momentum. I really can start to envision this, and you're getting the state labs that do the genetic screening getting much more engaged in these discussions. I don't think it's a matter of if. I think it's a matter of when, and I think it might be within striking distance.
And I guess so much of the science has been done. It is already being incorporated in medicine. So you already have that transition into the private sector, right?
Exactly.
And nothing's going to shut down that funding.
That's right. And you said something earlier. You sort of implied that we're getting close to $100 for a whole genome sequence. And we're getting close to that. Well, I want to remind you that right now, when you do newborn screening, as all state labs do for a few dozen genetic disorders, it varies by state, the price tag on that is like $50 to $75. So if it's $50 to $75 to get information about a few dozen genetic diseases, and for $100, you could get information about the over 6,000 rare genetic diseases for which we know what gene is mutated, I think the bargain is with the $100 test. And so now I'm oversimplifying it, and there's lots of barriers and lots of challenges and lots of things we're going to have to do. But within 10 years, I can't imagine it won't be the case. And I think we're going to see more and more.
You're bullish on it. You're bullish on it. That's what I've been calling it this year.
This year's, I think there was something about that UK announcement declaring they're doing this within 10 years, along with all these other developments. And by the way, if you follow the scientific literature, studies after studies and review article after review. I mean, there's just this huge amount of attention, and that's good attention, because I'm not saying any of this is simple, but it's incredible energy and incredible productive discussions, and including the ethical challenges and the literacy challenges and the logistical challenges. Lots and lots of things have to be solved before we do this, but I can start to see us being on this trajectory.
There's also some new companies, some exciting new things going on. Tempus is kind of exciting, I think. There's companies that are, there's a software company called Nest, which is helping providers, training them. It has a software to basically bring them on to genomic medicine. I think it's addressing.
I just recently met her, the CEO, and they're also doing
Miran,
there are a lot of really important things that they're also doing, such as online AI-assisted tools for getting people to do informed consent for participating in genomics research, or eventually for getting genomic testing done. So, you can start to see all these pieces and productive uses of AI, and, it just gets very exciting.
You could say the genome's already out of the bottle.
The genome, yep. No, you're absolutely right.
Well, they had that, Genome in a Bottle consortium for accuracy. Okay, cool. Well, it's exciting talking to you. And so the flavor of your talks, I should have asked you for one of your slide decks to prepare for this, but the flavor of your talks is that, hey, we got some real headwinds going on right now. There's some major disruption. But then to focus on just the incredible good news and progress that's also happening simultaneously.
Well, first of all, when I give talks, I'm enthusiastic, upbeat. I, occasionally will mention what happened to me, but I haven't even mentioned it. That's history. That's in the rear-view mirror.
Oh, okay. Just focus on what's happening.
When I'm giving my vision talk, it's all, yes, we've got challenges, but most of the challenges I talk about are the genomic challenges, because, and I do that very much in the spirit, because I don't want young investigators or trainees who are hearing me talk. They shouldn't be worrying about these big problems going on at NIH. Let the old people worry about that. And there's plenty of energy being put in to try to right the NIH. I want them just to keep their head down, focused on these incredible opportunities and the challenges that come with it, because we need to keep up that innovation. We need to keep up that intellectual curiosity and problem solving, because, genomic medicine is not going to happen without a lot of blood, sweat, and tears. And we need to continue to build that workforce. And it's a workforce of many different disciplines that are really going to be needed to see this fully realized.
Let's talk about where the science is headed. I think you had a unique position because you're in charge of that funding. Um, and I remember running into you a couple years ago at ASHG, and I was telling you, hey, I'm getting so many companies and guests on the program talking proteomics.
Yeah.
Are you taking some of that genomics money and putting it in proteomics? And you were like, they need to go to Congress and get more allocation. Um, but it is becoming more about multiomics. It's like we and now this week I interviewed CEO of a new company called Selenome. We'll just be publishing that soon. Their point is to get back to the level of the cell
Yeah.
and then quantify everything, not only genomics, but interactomics and also their imaging. So they're combining imaging with some of these molecular technologies. First of all, they stage the cell though and keep it alive, right? Because we've never been able to do the in vivo thing.
Right.
So what I've seen on the program is, we were at cell biology back in, the last century, 50s, 60s. Then it went all molecular, more reductive. Now it seems like it's coming back to the cell, but this time we have all that reductive knowledge about the molecules.
Right. And so, again, you're marrying that with a lot, and you're marrying that with a lot of computational power and a lot of AI to sort of help you put more complex pictures together of incredibly complicated choreography, I'm sure, that's going on in the cell. But by the way,
Yeah,
I want to extend on what you were saying
I was
about
hoping you'd have more to say.
my hesitation around proteomics, and because a couple things changed. So first of all, it was well known that I was always trying to keep us confined to genomics and not get too pulled into too many other omics. In part because NHGRI only has 1.4% of the NIH budget. We have this massive intellectual footprint, incredible excitement around genomics and other things omics, but we're teeny. And I always felt like, we can't allow too many other things to creep in to
I
dilute our
see.
genomics. However, I will say in the last few years that I was the director, we actually had a small technology development program to try to use nanopores to do protein sequencing, to sort of adapt some genomic technologies for doing proteomics. But more importantly, that I'm very proud of, and this is getting a lot of attention, and I might even suggest you might want to get some of these investigators and maybe get them on one of your
On the
podcasts,
show.
because this is
Okay.
a growing area. One of the last things that got launched before I got terminated was a project I was very excited about. Actually, it came out of our 2020 strategic plan, or strategic vision, very overtly. We talked about this multi-omics idea. You already mentioned the phrase. And so we launched a program. It's called Multi-Omics of Human Disease, or MOHD, MOHD. And every one of those investigators, what their requirement was to study a human disease or physiological process using genomics and then at least one or a couple other omics. And it was their choice. So yes, we were funding some other omics, but the whole idea was to take a multi-omic approach that would force data integration, data analysis, exactly what we know the future is going to be. It's almost what some of your cell biologists are going to be interested in doing, understanding how the cell functions. We were asking them how to study a disease area or a disease specifically using a multi-omic approach. And of course, we brought with us some other institutes who were interested in specific diseases for specific grants that got funded. So it's like about not quite a year old, maybe it is over a year old, I lose track of time now, but it's called the MOHD Consortium. You can look it up and see who the investigators are. And it is emblematic of a growing area of omic research or precision medicine, whatever you want to call it, because it's all about data integration. I'm a big fan of genomics, but I've never said genomics is everything. It's just the wave I've ridden for my professional career. We know we need other data, and it's going to be really relevant to integrate that data.
Right. And are you saying, and by the way, we had to get there with genomics. We had to characterize the genome to be able to even start thinking about, the RNA and the proteins.
And we've been fortunate. We're riding this technology surge. It was not a given that we were going to get as many wonderfully impressive technologies for DNA sequencing as we've had in the last 22 years since the end of the Human Genome Project. Now we're getting better proteomics methods, and then we're going to get better, metabolomic methods. So I've never said, I mean, I've just said that genomics has been, this incredibly exciting revolution, but we've been lucky. It's not all luck, but, we've been fortunate that the technology really was able to advance very quickly, very aggressively. And as other technologies, even that for measuring the environment around us, or for measuring better what we're eating or what we're being exposed to. When those technologies, and some of which are available even on our iPhone these days, when those technologies get more robust, you're going to see a revolution with that data as much as you saw a revolution with genomic data, especially when you put the data together.
You got there before me. So I interviewed two leaders. One directs the Nexus Center for Exposomics, Chirag Patel up at Harvard, and one of their points on funding. And this goes back to what you were saying earlier. You wanted to protect that 1 point whatever 4%, and keep it going to genomics. Doesn't mean that another institute, like one of the disease institutes, couldn't fund these proteomics or other omics
100%.
studies. And when I asked Chirag about, are they getting funding, because that could be the sign that this exposomics is maturing into a real field. He said, yeah, but, they were getting funding from several different institutes together, because they're all interested in the exposome.
That's right. That's right. So that's, that's often how NIH works is, for certain things, you'll make up multiple institutes together. And every one of them also are trying to preserve their core area and disease area or technology area.
Okay. So let's end with a little bit about the future. What's exciting you today? I mean, you've mentioned some of it here today, but what's exciting you, like looking 5-10 years out? Obviously, you're bullish on newborn screening for healthy individuals. What do you think should be the mission of the NHGRI moving forward? And do scientists need to be more aware of the politics somehow? I don't know, but to make that argument.
We talk. Let's start with the last thing first.
Okay.
The politics. I think the politics are really complicated, and when people ask me about the politics, I never know. Should I be coming at this from the point of view of a 31-year government official and what happened to me and what happened at NIH? Should I be coming at it from the point of view of an American who cares a lot about this country and who has been a political wonk his whole life? Or do I come at it from the point of view of a father who I feel like, what am I giving my kids are, quasi grown up at 29 and 26. But, in some ways, I feel guilty that we're having this country that seems to be falling apart to them. So I don't know, I'm more worried as a father or as an American or as a scientist. I worry about all of it. But, what I would say about the science in particular is that I think we should feel really good that Congress, bipartisan support, continues to support it. But it doesn't mean that we don't have to fight tooth and nail for this anti-science rhetoric that seems to be running rampant in too many people in this country. Why is it so many people in this country are as anti-science as they are? And these are people that, that we're living in our communities. They're the people that we're interacting with all the time. We need to be thinking about what we're doing wrong, that these people are being led astray. And I think we all have a responsibility. So I'm a very strong advocate that we all need to be part of the conversation with and directly try to find out from people, why don't you trust scientists? Why don't you trust what, some of these people who are, I think, are heroes are being demonized? Why is that? And by the way, I don't think the solution as scientists is to say, woe is me, I didn't get my grant. I don't think people are going to be sympathetic to that. I think the woe is me is, woe is me, I'm working on this multi-omics where I believe we will better understand how to deal with this rare genetic disease or this common disease. Like I heard your aunt has diabetes. We are getting more and more insights about the key molecular and environmental components of diabetes. And out of that, we're getting better drug treatments. Try to tell it at a patient story about, not about woe is me, I lost my grant, but woe is me, the science isn't getting done and it's beneficial. And
do that at dinner parties. Yeah, do that at dinner parties. Do it at the bar. Do it at gatherings with family.
Do that with your Uber driver. I've spoken to more Uber drivers trying to explain to them what the implications are of bad NIH. And then you explain to them the story of how we end up curing sickle cell by gene therapy. If you go through and tell that story, because some of it is some of the most basic science research that some people think is unimportant, and yet it teaches you things about how to do genetic engineering and genome editing, et cetera, et cetera. There are so many good stories to tell that really are saving lives now. So that's the political side of it. Now, what's exciting in genomics? I mean, I think what I would say more than anything is everything's full steam ahead with clinical implementation. It's not that there's not incredible Herculean efforts being made to better understand how the genome works and better understand genomic variation and all this basic science and translational science things. But what I'm finding gratifying, and maybe it's because I'm getting older and more sentimental, is I just never thought when I got involved in the Genome Project in 1990 that I would one day be able to say genomics is saving lives. I thought that was for the next generation because I didn't think it would come in my lifetime. I mean, we are saving lives. I mean, there's just so many examples of this now, and we really are improving the lives of people with undiagnosed conditions that we're getting them diagnoses. In some cases, we're getting them treatments. It just seems that more and more of these things are coming up. That and then the prospect that, again, maybe in the next 10 years, we may end up in a circumstance where in an open, equitable way, the way genetic screening is done now, all newborns coming into the world will have a genome sequence that will enable their medical care from the time they are born. That's like breathtaking. And, I know it's complicated, and I know there's lots of major obstacles.
And we get into those on the program. We do.
I mean, but that is not, it's no longer something for the next generation. It's for this generation. And that just makes me very excited. And to see all this effort in getting to the point of making genomic diagnoses then being met with therapeutic options, colleagues that I watch from afar because I don't take any credit in the therapeutic space, but they're miraculous, and it's very gratifying to see.
What do you want to do?
What do I want to do?
Ideally.
I think I'm doing it. Even
giving talks, being an advocate.
I consider myself, I'm finding I really love, because some people say, oh, are you going to go back and start a lab again? No, I really like being a genomics evangelist. I really like being an advocate. I really like giving career advice, giving institutional advice, giving programmatic advice. I really find that I'm a problem solver. I like to be very upbeat, positive, and evangelical, but I also like problems. I mean, I like to say, why is it, give me a roadblock. Why is it that this is not being solved? Give me that's why I've started talking to some, state public health labs. What are the obstacles you see to being able to start sequencing genomes?
Awesome.
That's what I sort of see, and exactly what form that takes and where it takes and all that. But I love teaching. I love doing these kind of communication. I love doing the outreach, and I love sort of trying to help identify the problems and trying to do problem solving. I'm doing it at a higher level. I'm not going to run my lab. I'm not going to run a research program, but I still think I can be helpful.
What did you learn about yourself this year?
What did I learn about myself is I think I always considered myself resilient, but I certainly came to learn that when bad things happened to me, I quickly felt a lot more sorry for a lot of other people than me. And I still feel really bad for people at my institute who I miss terribly and they miss me terribly. And oh, by the way, I didn't say that, and maybe that is a nice way to put the last two things I've talked about together. One thing that I did learn about myself is that I know is absolutely true, and it's a little bit about what does my future hold, is I am officially on call anytime to help rebuild the NIH. And I don't know when that time will come. I'm hoping it'll come in 3 years. I don't know what that's going to look like. I don't know what form it'll be. And it could just be giving advice. But there will be a time where we need to do a lot of damage. It's really easy to destroy a place. And we're in the middle of it.
And very hard to build it. Yes.
It's like watching a house get rebuilt, right? They can rip a house down in an afternoon, and then it takes 4 months to rebuild it. It's going to take a long time to rebuild. And I don't even know where, right now I'm living in Atlanta most of the time. And I don't know where I'm going to be living, but there's one thing I'm not doing, is I am not selling my house of 31 years that's located within a 5-minute drive of the NIH, because I am on call and I'm going to keep that house ready. And I'll visit it frequently. I may spend time there, but I am officially on call that when NIH goes into a rebuilding phase, they can call me and I'm going to have a house to live in and help them in any way I possibly can.
Eric Green, former director of NHGRI, thank you so much for coming on our program.
Well, it's been wonderful talking to you, and happy holidays to you and your entire audience.